Thank you very much for the nice introduction, Barbara, and thanks to Dennis and Barbara for inviting me to present this afternoon and to everyone for listening in. So my name is Will Norton. I'm an Associate Professor of Animal Biology in the University of Leicester, that's in the United Kingdom. And today I'm going to tell you about two stories out of my lab which look at the genetic and pharmacological basis of zebrafish aggression. Research in my laboratory is focused for quite a long time on trying to understand how animals produce and control aggression using zebrafish. And we have projects where we identify novel aggression-linked genes, when we analyze genes linked to human psychiatric disorders that include aggression as a symptom, such as ADHD and autism. And we've also recently been screening for novel drugs that can reduce aggression. My lab also has projects looking at the release of neurotransmitters in the brain. We use fast-scan cyclic voltammetry, and I have a PhD student called Palumi who's very interested in dopamine and is currently characterizing a dopamine transporter mutant. And finally, we're also interested in fish welfare, and I have a postdoc called Ken Wynn who's trying to improve DNA collection methods in both zebrafish and stickleback. But today, I'll be trying to talk about a simple question which my lab has studied for 10 years or more, and I'm not sure how much closer we are to really understanding this. And what we want to understand is what makes an animal aggressive. So on my slide here, I've depicted a cartoon showing the zebrafish brain. You have the forebrain here, the optic tectum. We have the hindbrain going down to the spinal cord. And you can imagine that a fish may be approached by another animal. It could be a conspecific, so another zebrafish, and it has to make a decision whether or not to be aggressive or to retreat. And so that would involve visual inputs. They have to notice the conspecific, processing within the optic tectum, processing within the forebrain where there are nuclei such as the amygdala or the homolog in zebrafish, which gives valence to the stimulus to say whether it's aversive or not. And then the animal has to tailor an appropriate behavioral output using motor circuits. Aggression is a really important adaptive behavior that animals use to protect themselves and their offspring, to fight for food or territory, and to establish social hierarchies. However, the levels of this behavior need to be tightly controlled because heightened aggression can lead to injury to the animal or even death. So ultimately, what I'd like to understand is how do animals make the decision whether or not to be aggressive? And how does the brain process this information so that the zebrafish can tailor this appropriate behavioral response? So one of the reasons I started studying aggression in zebrafish is that it's relatively easy to measure. You can either place two fish in a tank and look at their aggressive interactions, depicted on the left-hand side here. And this is a cartoon which I've taken from a paper by Rui Oliveira and colleagues. And in these dyadic fights, zebrafish will show characteristic agonistic postures, which will include sizing each other up, biting, moving the tail very rapidly to fight. The other way in which you can measure aggression is to simply look at interaction with a mirror. And I have a very simple video on the right-hand side of my slide here. And this shows our standard aggression setup. You have a tank with two white opaque walls, which aren't reflective, and we placed a mirror outside of the tank. And you can simply record the fish's aggressive interactions with its own mirror image. A zebrafish can't recognize itself in the mirror, so once again it will show these characteristic agonistic postures, including biting against the mirror and pushing, and thrashing the caudal tail very rapidly. The mirror-induced aggression doesn't recreate all aspects of a dyadic fight. For example, we know there are some hormonal differences between these two paradigms, but we do see a good correlation of aggression levels in both setups. And classically in my lab, we tend to use this mirror stimulation protocol because it's a refinement and a reduction, meaning that we can use less zebrafish and they're less likely to hurt each other. When I started doing this work, I was a postdoc in Larval Behavior lab in Munich and then in Givry in France. And we were lucky in that we were able to identify a zebrafish mutant line, the FGF receptor 1A mutant line, which showed heightened aggression compared to wild type. So this is one of the figures from what's now quite an old paper from 2011, where we were able to show that the FGF receptor 1A mutants are more aggressive than wild type. That's depicted here. And this is an experiment where we simply placed either wild type or FGF receptor 1A mutant fish into our setup and recorded their interaction with the mirror for 5 minutes. And you can see that the Spiegel are more aggressive than wild type. And interestingly, the Spiegel or the FGF receptor 1A mutant fish maintain high aggression levels for a long time period, whereas the wild type fish reduce aggression levels over time in a 30-minute experiment. So this was tremendously exciting, and it enabled us to identify another signaling pathway, FGF signaling, which is important for the control of aggression. And by looking at the brain areas which express FGF receptor 1A, it gave us some ideas about which areas of the zebrafish brain might be important to control aggression. And it's a somewhat classic approach used by a lot of zebrafish labs to create a mutant line and then study the phenotype. But I think one of the worries is that a mutant line may suffer from genetic compensation over time or redundancy. You could have several genes which undertake the same function within the animal. So perhaps studying these mutant lines don't give you so much information about the behavior of a wild-type animal. And so in a recent study, we've been using drugs to do similar experiments, but the drug approach is also very nice, the pharmacological approach, because if you have a well-characterized drug, you can acutely manipulate the wild-type brain for a very short time period. And once again, you can then try and understand the signaling pathways or brain areas which have been activated. So in my talk today, I'm going to try and show you two short stories, and hopefully I won't confuse you by switching between the two. First, I'm going to tell you about screening for novel drugs in zebrafish, and I'll focus on caffeine and sildenafil, two targets we identified. I'll then show you some unpublished data about a histamine receptor 3 mutant line, and then in the final slide, I'll try and pull it all together and talk a bit more about the neural circuit in the zebrafish brain which controls aggression. It's something I'm very excited about, so hopefully I can convey some of this excitement to you today. And so the first project I'd like to talk about, we were a part of an EU-wide consortium called Aggressitype, which aimed to better subtype aggression in humans and other animals, with a view to improving treatments available for human patients which exhibit high aggression levels. So in humans, aggression is a symptom of several psychiatric disorders, including attention deficit hyperactivity disorder, that's ADHD, and conduct disorder. However, the drug treatments available to reduce or control aggression are poor at the moment, and in some cases they have unwanted side effects. And there's a real need to identify novel drugs which can be used to control aggression levels. The zebrafish is an ideal model for drug screens because they're relatively small, they develop quickly, and drugs can be applied by immersion, so simply by dissolving the drug in the fish's tank water. And this is an approach which has been used beautifully by many zebrafish labs, including Jason Reel and Dave Prober, for example, where they looked at sleep behavior. And so we reasoned that using our mirror-induced aggression setup, we may be able to screen for novel drugs which can reduce aggression levels. When we started this project, we were lucky that we were able to interact with a behavior software company called Viewpoint, and Viewpoint built us a bespoke setup to measure juvenile zebrafish aggression. Previous research in my lab had already shown that zebrafish exhibit aggression from about 4 weeks onwards. So if you take a wild-type zebrafish and you have a group of them, at 3 weeks, some of the animals are mature enough to show aggressive interaction with the mirror, but by 4 weeks, nearly all of the fish in the group show this aggressive interaction. And we thought that the juvenile stage would be really useful because once again, they're smaller than adults. It's quicker to generate them in large numbers, and we could record the behavior of more juveniles in parallel compared to adult animals. And so we used these 1-month-old juvenile fish in our screen. The novel setup that Viewpoint built for us allowed us to record the behavior of 12 animals in parallel. It's depicted on the left-hand side here. This is Arnie, the aggressionator. And you can see that we have 3 cameras in the top and we got our 12 arenas underneath. And our setups were designed to be a miniature version of the setup we use for adult fish. So the tank has 3 white opaque walls and there's a mirror placed outside to elicit agonistic behavior. We also had a white slider we could place in front of this tank, and that meant that we could first immerse the fish in the drug, so that's depicted in panel B here. We could then place them in the arena with the slider present and record locomotion without interaction for a minute with the mirror for 30 minutes, and we could then remove the slider and measure aggressive interaction for 30 minutes. So in a proof of principle study, we simply wanted to show that we were able to detect aggression using this setup and also detect changes caused by applying drugs. So we used a sort of reverse translational approach whereby we took drugs which we knew would alter aggression in humans, we applied them to our larval zebrafish. On the top 2 panels of this slide, you can see we have lithium carbonate, which is a drug which is used to treat schizophrenia. And in 1-month-old fish, lithium reduced aggression levels at the highest concentration we used, 5 micromolar, and it also caused a reduction in locomotion at this concentration of drug. Conversely, we applied ethanol to our juvenile fish, And at the highest concentration of ethanol that we used, we saw an increase in aggression without affecting locomotion. And so that showed the software we had and the setup we had is able to discriminate aggression levels between animals with and without application of drugs. And it meant that we could use this within our drug screen. Over the course of the next year, we screened nearly 100 drugs for their ability to alter aggression and/or locomotion. And we designed a phenotype-based screen to examine drugs that reduced aggression but did not affect other behaviors. So we wanted them to be as selective as possible for aggression. And we decided to do a bias screen based upon drugs identified in the literature. And we chose to do this because actually we could only look at a relatively small number of drugs, 94 within the year time that we had. We want to maximize our chances of finding positive hits, or a reduction of aggression in this case. We screened each drug at 3 different concentrations on the same day, and we applied them by immersion acutely for 30 minutes. And we then used sedation or a lack of swimming as a readout of drug effect. So if we saw that a drug reduced aggression, but it also reduced swimming in the tank, we didn't find it a very interesting candidate. The way in which we designed this screen was in a 3-tier process. Initially, we applied our drugs to a rather low number of animals, only 10 zebrafish each, and we looked for changes in aggression, and we weren't too worried about significant changes in behavior. By doing this, we identified 30 promising compounds, which we then retested in a second tier of the screen. And in the second case, we applied the drug to 30 different zebrafish and remeasured their behavior. And by doing this, we were able to get rid of some false positives. And we identified 5 compounds which significantly altered behavior compared to control-treated animals. And finally, we selected 2 of these for further study to look at their activity in detail. And we applied these to 100 fish in each case. We got very robust statistical results for caffeine and sildenafil. And so in my next few slides, I'll simply walk you through the data that we have first for caffeine and then sildenafil. Caffeine, as you probably know, is a methylxanthine derivative, which is very commonly found in foodstuffs and drinks. It's the most commonly used psychoactive drug in the world, and the concentrations in which we encounter it in our diet, it acts as an antagonist of adenosine A1 and A2 receptors. So we found in the first round of our screen, using a large number of larval zebrafish, that 100 micromolar caffeine could reduce aggression compared to control-treated animals without affecting their locomotion. And actually, we were quite surprised about this. Initially, a priori, we thought that if you drink a lot of coffee, for example, it might make you more nervous and agitated and more aggressive. And actually, we found that in this animal species, we saw a reduction of aggression using caffeine. And that's probably because we used a very low concentration of this drug in our screen, only 100 micromolar. So we saw a nice reduction of aggression in our larval fish. What happens in adult animals? So in this case, we simply repeated our experiment using adult zebrafish. And in this case, we used a larger number of different concentrations of the drug. And actually, you can see that using adult zebrafish with this acute immersion and that adult setup, we saw a very strong reduction in aggression levels, which is much stronger than that which we'd seen in the juvenile animals. In parallel, we saw that locomotion was only affected at some of the doses we used, but not all of them. And we saw that by and large, most of the animals which were immersed in caffeine spent a similar amount of time interacting with the mirror. Only when we used the highest concentration using the screen, 200 micromolar, did the animals no longer interact with the mirror. So this was a really striking result. Using 100 micromolar caffeine, for example, we see this very dramatic reduction in aggression levels, and we haven't affected locomotion, or we affect locomotion to a lesser extent. So as I've already mentioned, we know from other species that if you drink a lot of coffee, it can make you anxious, and caffeine is known to be anxiogenic if given at high concentrations. So we wanted to know whether the low concentration we'd used to reduce aggression in our screen also altered anxiety. And in order to look at this, we used the novel tank diving test, which has been very extensively used in adult and juvenile fish. And this simply looks for a thigmataxis, which is the preference for an animal to spend time at the bottom or the side of an arena. And a more anxious wild-type fish will spend more time at the bottom of the arena, whereas a less anxious fish will swim up towards the top. At the concentration of caffeine which we used in our screen, we saw that animals spent a similar amount of time in the top of the tank compared to control treatment. There was no change in the amount of time they spent freezing, There was no alteration in the total distance swum by these animals. So at the low concentration, which reduces aggression, caffeine does not appear to be anxiogenic. We next went on and measured cognitive impulsivity using the 5-choice serial reaction time task. And these were experiments which were done by Matt Parker at the University of Portsmouth, who has really done a lot of work setting up this 5-choice test in zebrafish. And the 5-choice test is very elegant. It's a test which was set up for rodents, and it allows you to look at different aspects of cognitive impulsivity. And the 5-choice test is depicted on my slide here. So we have an arena with 5 different choice areas, each of which contains an LED. And on the other side of the tank, we have a food hopper, which can give a food reward. The 5-choice serial reaction time task is a 2-step experiment. In the first step, you have Pavlovian conditioning, where a zebrafish has to learn to pair the illumination of an LED with a food reward. So when the LED is switched on, if it nose pokes the correct LED, the fish can then go and correct its food reward. So that's a simple learning paradigm, and zebrafish are very good at doing this. In a second step, you introduce a gap between the light being illuminated and the availability of the food reward. It's called an intertrial interval, an ITI, and in our case, we used a 10-second intertrial interval. And so in the experiment, only one of the 5 LEDs is illuminated. The fish then has to wait 5 seconds before nose poking, and it can then go and collect its food reward. So you can look at incorrect responses, which is nose poking, the incorrect LED, which isn't illuminated. And you can also measure animals which are unable to wait the 10 seconds before nose poking as a measure of impulsivity. So when we applied caffeine at the doses which we'd used to reduce aggression, we saw a decrease in the number of correct responses in this test, which reaches significance at 50 micromolar. So that meant that the adult fish nose poked the wrong non-illuminated LED. And we also saw a significant increase in the number of premature responses. So when we apply caffeine, it appears to make these adult fish more impulsive. So this is a very interesting result because one of the big aims of the Aggressor Type Consortium was to try and understand to what extent we can separate impulsivity and aggression out as 2 different behavioural phenotypes. In some animal species, such as humans, you have 2 different subtypes of aggression, which can be predatory, which doesn't include impulsivity, or offensive aggression, which is impulsive. So you can divide aggression into 2 subtypes depending upon the level of impulsivity. So it was interesting to see that when we altered aggression in zebrafish, we also altered impulsivity. So this was a really cool result. So we'd already found a really promising drug which can reduce aggression without affecting anxiety, but increases impulsivity. And we next wanted to understand how is this drug active in the zebrafish brain. And in order to do this, we used neural activity imaging using GCAMP6S, which is a calcium indicator. And these studies were done in collaboration with Matt Winter at the University of Exeter. And in this case, we used 6-day-old fish because the brain is much more opaque at that stage. So what Matt did was he either applied a control treatment or 100 micromolar of caffeine for 30 minutes, and then he placed this fish which expressed GCAMP6S under the ELAV3 promoter, so pan-neuronal promoter, within a light sheet microscope, and recorded calcium activity. On the left-hand side, we can see control calcium activity, so the fish is being moved, rotated within the microscope, so we have some background activity. And on the right-hand side, we can see what happens if you apply 100 micromolar caffeine. So this answers the endless question, what would happen if I give my goldfish coffee? And you can see it doesn't play particularly well. I'm afraid my computer's struggling here at home, but hopefully you can see activity in some areas of the brain and some large flashes of activity in the hindbrain here. I'll just let that run. So there's many ways in which you can quantify this type of imaging data, and you can look for significant changes in different brain areas between the control treatment and the animal which has received the drug. I won't show you this data in detail for the sake of time today, but what we saw was that when we applied caffeine to our larval zebrafish, we saw significant changes in activation of the eminentia granularis, the tangential vestibular nucleus, and the valvular cerebellum. And interestingly, these all form part of the larger cerebellar area of the zebrafish. On the right-hand slide here, I have a picture depicting the adult zebrafish cerebellum. And here you can see, here's the cerebellum with its Purkinje cell layer, molecular layer, and granule cell layer. And you can see the position of the eminentia granularis and the valvula. So initially we were quite surprised about this, but when we looked at the literature, there are some recent studies in humans that show that when we drink coffee, the cerebellum is actually activated, and so that fits with the data which we were able to recover using our zebrafish. So in summary, for our first target, we looked at caffeine and we saw significant activation of the cerebellum, and it seems to be important to reduce aggression at both juvenile, and adult stages. The second drug which we chose to look at in detail is sildenafil. And sildenafil is a potent and selective phosphodiesterase 5 inhibitor. It has a trade name, it's already given to humans, it's called Viagra, and it's used to treat both pulmonary hypertension and erectile dysfunction. And importantly, phosphodiesterase 5, the target for sildenafil, is only expressed in one type of neuron in the brain, and that's within Purkinje cells of the cerebellum. So once again, the cerebellum appears to be important for the activity of this drug, and as I'm about to show you, for the reduction of aggression. So using our juvenile 1-month-old fish, we did our screen data. In the third round of the screen, we were able to show that 10 micromolar sildenafil can reduce juvenile zebrafish aggression, depicted here, without affecting locomotion. We also looked at whether sildenafil is active in adult fish, and once again we saw a strong reduction of sildenafil in the medium concentrations, strong reduction of aggression in the medium concentrations of the drug that we applied. So at 1, 5, and 10 micromolar, we see a reduction in zebrafish aggression, but not at the highest concentration, 50 micromolar. And in this case, we don't alter the amount of time that the zebrafish spends in the mirror, and nor do we alter the zebrafish's locomotion. So it seems to be quite a selective reduction of this behavior. We also did the 5-choice serial reaction time task. I won't show you the data today because I'm worried I'm going to run out of time. And we saw the opposite phenotype. So sildenafil makes adult zebrafish less impulsive compared to caffeine. And this is absolutely fascinating because we've taken two drugs, both of which can reduce aggression, but we've shown that we can, in parallel, alter impulsivity in the opposite direction. So we can actually separate out these two behaviors in this species. Finally, we went back to Exeter again and asked Matt if he would be so kind as to image the whole brain using GCAP success when we apply sildenafil. And on the top here, we have our control brains. You've already seen this film. On the bottom, you can see the areas of the brain which are activated by sildenafil. Great, my computer's a bit slow, but you can see it's working there. I'll just leave it running in the background. And in this case, we actually only saw significant activation of one area of the 6-day-old larval brain, and that's an area called the eminentia thalami, which interestingly is thought to link Purkinje cells in the cerebellum to the forebrain. So that might explain why you have phosphodiesterase 5, this target expressed in the cerebellum, and why we see activation of this thalamic nucleus, the eminentia thalami. And so in the first part of my talk, I've given you a very brief run through of a drug screen in which we developed a phenotype-based screen to identify novel drugs to reduce aggression. We screened 94 drugs and found 5 promising hits, and we looked at 2 of these in more detail, caffeine and sildenafil. Both caffeine and sildenafil reduce aggression in juvenile and adult fish, and we found that caffeine increases impulsivity in the 5-choice serial reaction time task, whereas sildenafil decreases impulsivity. Using calcium imaging in larval fish, we found that caffeine activates the cerebellum and sildenafil activates the eminentia thalami. And this is work which we published in a paper which has just come out at the beginning of this year. We think that it would be nice to try and develop these compounds into possible treatment options for human patients. Caffeine possibly isn't the best treatment in which you can use because it's used so widespread by many people across the earth. It's very hard to find people who aren't exposed to caffeine either by drinking or eating something in their diet, so it's not that great a drug. But I think that sildenafil and possibly other phosphodiesterase inhibitors could be really useful in this regard, so that's something we'd like to look into in more detail. So in the second section of my talk, I'm going to switch gears and tell you about a novel histamine receptor 3 mutant line. And this is work which was started by Amir Al-Usta, who is a postdoc in my lab, who set up CRISPR-Cas9 mutagenesis. He made a CRISPR knockout histamine receptor 3 mutant line for us. And then another postdoc, Florian Reichmann, took this line and he's done a beautiful job at characterizing this. Florian has now moved to the University of Graz to set up his own lab, so that's the first spin-off lab from mine. I'm really proud of this, and we expect great things from Florian. So if you remember from the very old paper from 2011 from Larval Behavior's lab, we'd characterized the FGF receptor 1A mutant line and shown that they were more aggressive. And we were able to locate the area of the brain which appeared to be important for this phenotype to an area of the hypothalamus called the Paraventricular Nucleus. And what you can see here are different coronal sections throughout the adult zebrafish brain. I have a cartoon of the brain here. The sections are about midway through, and we're focusing on the ventral portion of the hypothalamus. And within these two kind of feature-shaped protuberances, you have a donut-shaped nucleus called the paraventricular nucleus. In our mutant, we saw that this was atrophied in mutants compared to wild type. My cursor keeps disappearing here. You can see that here compared to wild type. And in other experiments I won't show you today, we found that histaminergic neurotransmitter signaling was important for the phenotype of these FGFR1A mutants. So they had a decreased level of histamine in the brain, and if we could rectify the histamine level, for example, by using an agonist of the histamine receptor 3, we could rescue their behavior phenotype. And that got us interested in studying histamine as an important neurotransmitter. It's been relatively understudied compared to other classic neurotransmitters such as dopamine or 5-HT, and there's very little known about the connection between histamine signaling and aggression. And so Amir went ahead and he set up CRISPR-Cas9 mutagenesis in the lab, and he was able to create a novel HRH3, so histamine receptor 3 mutant line, which has a 4 base pair insertion within exon 1 of the gene and reduces about 75% of the protein because we have a premature stop codon, so we think we have a full knockout. So in the first set of experiments, Florian simply looked at the areas of the adult zebrafish brain which express this histamine receptor 3. And what you can see here are coronal sections throughout the adult zebrafish brain with in situ expression in blue. And you can see that there's widespread expression throughout the brain in keeping with what we know about this receptor from other species, that it can act as both an autoreceptor and a heteroreceptor. So it can modify both histamine signaling and the signaling of other neurotransmitter pathways. And you can see strong expression in the telencephalon here, in the medial, posterior, and lateral parts of the dorsal telencephalon and some of the ventral telencephalon. Or posteriorly, there's strong expression in the preoptic area. And more posteriorly again, we see expression in the grey matter, the PGZ, the central area of the hypothalamus, which is sometimes called the nucleus of the lateral recess, and also within this periventricular nucleus area, which was important in the FGF receptor 1A mutant. And so HRH3 is expressed throughout the zebrafish brain, including some areas which we know to be important for the control of aggression, including the PVN, the preoptic area, and also the DL, which we think is the zebrafish homologue of the amygdala, so it's important to give valence to stimuli. So in the first set of experiments, we simply did our classic mirror-induced aggression depicted here, and what we saw was a strong reduction of aggression in our mutant fish, so that's the squares here, compared to the wild-type animals when we simply looked at aggressive interaction with the mirror over a 25-minute time period. We also wanted to see if other behaviours were changed in parallel. So we used an open field test depicted here. It should probably be called an open tank test really, but we always called it the open field test after the rodent test. And in this case, we simply have a large aquarium with white opaque material around the sides. And we look at the distance swum and the speed of swimming and preference to be in the center or the outside of this arena. And in this case, we saw that our homozygous mutants swam slightly faster compared to wild type. They swam slightly further compared to wild type, but spent a similar amount of time being immobile. They have a similar angular velocity, which tells you how many times they double back upon themselves when swimming. And they spend a similar amount of time in the center and entering the center zone. So that suggests that they're slightly hyperactive in this test, which could perhaps be indicative of anxiety, but it's not the strongest test for anxiety, really. In the next set of experiments, we used a novel tank diving test once again as a measurement of anxiety-like behavior. And I depicted the tank on the right-hand side here, and you'll remember that an anxious fish will show a thick metaxis, which is a preference to remain in the bottom of the tank and not swim up towards the top. And when we compared our mutant fish in this setup to wild type once again, we saw a strong anxiety-like phenotype. So there was a reduction in the amount of time spent in the top and the middle of the tank, and a strong increase in the amount of time spent in the bottom of the tank, which makes it look very much like an anxiety-like phenotype. We also looked at latency to enter these different zones, so latency to enter the top zone, the number of entries in the top zone, the total distance swum, And we saw significant changes here as well. So they took a longer time to enter the top zone, as you might understand, because we're at the bottom most of the time. And they also swim further and a little bit faster in this test. So it seems that the adult fish, which lack histamine receptor 3 function, show a reduction of aggression and an increase of anxiety-like behaviour. So once again, we wanted to understand which are the brain areas which underpin this phenotype. And in this case, we couldn't use the whole imaging and neural activity readout in the adult brain because it's opaque, so it's very hard to do that experiment using a light sheet microscope. So in this case, we used an immediate early marker called RPS6. And RPS6 is a ribosomal protein which is expressed when neurons are activated. So you can simply allow an animal to perform a behavior for a short period of time. In this case, we used 1 hour. You can then collect the brain and use an antibody against RPS6 to see which neurons are activated. So that will give you some idea of the brain areas which have been activated as a readout of this behavior. And RPS6 has already been used in mouse, so it works very nicely. And what's nice about this marker is that you can use a pull-down assay followed by RNA-seq to then identify activated neuronal markers within these brain areas. So we've already used RPS6 to identify the brain areas which are important for mirror-induced aggression. I'm about to show you that. We haven't done the pull-down experiment yet, which would be really cool. So that's something we'd like to do in the future, and perhaps identify novel markers which are activated in response to an aggressive interlude. So the first step, we wanted to double check the phenotype of our mutants. And once again, you've seen this data already. So using somewhat smaller arenas, we see the same phenotype, which are histamine receptor 3 mutant fish are less aggressive. These are the squares, compared to a wild-type fish in the same arena. These are the circles. And we allowed them to sit in this tank and fight a mirror for 1 hour. We then collected their brain and used immunohistochemistry against RPS6. And you can see the results here. So on this slide, you can see activation of the telencephalon. We have 4 panels, 4 sections of brains from different fish. On the top left, we have a wild-type animal which was placed in the aggression setup, but there was no mirror present, so there was no aggressive interaction. And on the right-hand side at the top, you can see the same experiment using our histamine receptor 3 mutant fish. And then on the bottom, we compared this to the same areas for animals which have undergone this aggression experiment. So you can see on the top, we have the background expression of RPS6, and there's very little expression. These brown dots are neurons which express the immediate early marker. And on the bottom here, you can see reaction of a wild-type fish brain to an aggressive interaction with the mirror. And you can see strong activation within the telencephalon, and you see similar areas are also activated within the mutant brain. So this is the ventral part of the ventral telencephalon, and this is the medial part of the dorsal telencephalon. However, excitingly, we see differences in activation between the mutant fish, so that's bottom right here, and the wild-type animals after the aggressive incidents. We see fewer cells are activated in the VV area in the mutants, and we see more cells activated in DM compared to wild type. So that's up here. On the next slide, we also looked at other areas of the brain which were prominently activated, and we focused on the hypothalamus because this is the area of the brain which expresses histaminergic neurons, and we already know it's important from our FGF receptor 1A mutant line. So on the top, we have our control experiments for wild type and HRH3 mutants. You can see very little staining. And on the bottom, you can see that aggressive interaction with the mirror caused activation of neurons within the central nucleus of the hypothalamus, also called the posterior recess. And we actually see stronger activation of this area in mutants. So this is very exciting for 2 reasons. Firstly, we don't have a lot of information, or we still don't have a lot of information about areas of the adult wild-type brain which control aggression. So this gives us some pointers about some of the brain areas which seem to be important. And perhaps unsurprisingly, aggression seems to involve multiple areas throughout the telencephalon and the hypothalamus. And then by comparing to mutants, we can look at differences between the staining pattern, and that might give us some information about some of the brain areas which are mediating this reduced aggression, heightened anxiety phenotype of the histamine receptor 3 mutants. Okay, so we've quantified this data. This is simply cell counting from the boxes shown on the slides which you just saw previously. And you can see, for example, that aggressive interaction with a mirror in the telencephalon activates the lateral, the medial, the ventral, and the dorsal parts of the telencephalon. And intriguingly, we see heightened activation of this area in the mutant fish, so that's depicted here compared to wild type. after the mirror fight, and we see reduced activation of VV, so the ventral part of the telencephalon in mutants compared to wild types. And we also quantify different areas of the hypothalamus, so the central hypothalamus, the posterior recess, the ventral hypothalamus, which is also PVN, lateral, and the preoptic area. And in this case, once again, the aggressive interaction activates all these areas of the hypothalamus, but we see more or greater labeling of RPS6 in the HRH3 mutants compared to wild type. So it seems to be overactivated compared to the wild type fish. Finally, in the last few experiments I'd like to show you today, we also looked at larval behavior. And these are experiments which have been done by Neil Rimmer, who's a PhD student with Joe McDermid, and Joe's a zebrafish electrophysiologist who has a lab next door to mine in Leicester. So it's really nice to talk and collaborate with him. And Neil was kind enough to let us use and help us use this larval behavior setup he's made for 4-day-old fish. So we wanted simply to compare the behavior of the larval fish to our adult phenotype and to see to what extent is it similar. And we used different tests, including unstimulated locomotion in a Petri dish. We looked at a light-dark test where we looked for preference to stay on the light or dark side of the tank, and a startle test with a looming stimulus to drive the startle. And in all of the tests, we saw a very similar phenotype, which is a strong reduction of locomotion in the HRH3 mutant fish compared to wild type, at an increased amount of time that the mutants spend immobile, and a decreased velocity. And so it's very hard to interpret reductions of locomotion in a larval fish because, of course, it can be related to a lot of different behaviors. We wonder whether this is also an anxiety-like phenotype, because that would fit very well with some of the information which we've seen in adult animals, although, of course, the adult animals were a little bit hyperactive. So we still perhaps need to think a bit about how we put those two bits of data together. And finally, we went down to Exeter again and pleaded with Matt Winter, and we used the calcium imaging with GCAMP success to simply compare the brain of 6-day-old HRH3 mutant and wild-type animals in the light sheet setup. And in this case, we saw significant differences between wild-type and histamine mutants in the interpeduncular nucleus, the eminentia granularis, the lobus caudalis cerebelli, and we saw lesser changes in the eminentia thalami, the inferior olive, intermediate thalamus, tangential vestibular nucleus, and lateral recess. So some of these areas I've highlighted in bold, and that's because these are the same areas which we'd identified when using our drug screen. And we're actually very excited about this. So we've used these two different approaches, which are very different to each other, either acute manipulation of the wild-type brain using a drug, or characterizing of a long-term knockout in a fish. And in both cases, we've seen that very similar brain areas appear to be activated. So in summary, in the second part of my talk, I've shown you that we have characterized a histamine receptor 3 mutant zebrafish. I've only shown you some of the data, unfortunately, but I've shown you that adult histamine 3 mutants are less aggressive and more anxious than wild-type conspecifics. We also looked at larval fish, which showed that reduction of locomotion. In larval fish, we showed an impaired activation of the eminentia thalami, interpeduncular nucleus, and cerebellum, among other areas. And then in wild-type fish, we used RPS6 as an immediate early marker, and we saw neural activation of multiple brain areas by aggressive interaction in wild-type, including many areas in both the hypothalamus and the telencephalon. And we saw more distinct RPS6 labeling in the dorsomedial telencephalon and the central area of the hypothalamus in the HRH3 mutants, and reduced labeling in the ventral part of the hypothalamus. So this work is about to be submitted for publication. Florian has just done a really good job in writing up the paper, and we'll submit that, and we hope to get some good reviewers' comments. So for the future plans for the lab, as you've already seen, we have quite a few projects ongoing, but I'm very excited about this idea that I started off by telling you that I want to understand how does the brain process information to tailor a proper aggressive response to a stimulus, whether that's another fish, for example. And I think that converging evidence from several projects in our lab have independently identified areas of the brain, or the same areas of the brain. And I think it's going to be really fascinating to look at two of these in more detail. The first area, which would be very obvious to look at, is the cerebellum, particularly the Purkinje cell of the cerebellum. So the Purkinje cell was activated by caffeine in our imaging data. It's also the area where phosphodiesterase 5 is expressed. That's the target for sildenafil. We also want to look at the eminentia thalami in more detail. In the adult fish, the eminentia thalami gives rise to a nucleus, which is called the ventral entopeduncular nucleus. It's depicted here. And in some other vertebrate species, this is called the internal capsule of the globus pallidus. It's part of the basal ganglia. There's some really beautiful zebrafish behavioural studies which have already been published by people such as Christina Lilazar, we have Hitoshi Okamoto and Monica Folgueira. And if you read some of their papers, you can begin to connect together different areas of the zebrafish brain which might be important to control this behaviour. So, for example, we believe that visual information probably passes from the retina to the thalamus. That somehow activates the Purkinje cell in the cerebellum, but we're not quite sure how this works yet. The Purkinje cell outputs via the uridendroid cells, so that's the single output cell in zebrafish, and somehow it activates the ventral entopeduncular nucleus. The entopeduncular nucleus projects to the ventral habenula. There's beautiful tracing studies showing this, and work by Chu and colleagues in Hitoshi Okamoto's lab has shown that the ventral habenula is important for the control of aggression, and it interacts with the median raphe, which is the major source of 5-HT serotonin signaling, the zebrafish brain. And there's very well-known links between 5-HT and aggression. So at the moment, what I have is this beautiful idea, which is based upon basically a lot of hand waving. But I think I have enough information to really go and look at these two different brain areas in detail. We've already identified selective markers, which are expressed in Purkinje cell, thanks to work in many labs who've studied this cell type over the years. And we're currently trying to identify markers which are expressed in the ventral entopeduncular nucleus. And our aim is to develop chemogenetic or optogenetic tools which will enable us to alter the activity of these cell types or brain areas in real time, and then look at how that changes aggressive behavior. So my aim is firstly to try and get a PhD student to help me build some of these tools which we use, and then we can build a much stronger foundation and dig into this in more detail. I hope it's going to be really exciting. So that's all I'd like to tell you today, and thank you very much for listening. I'm sorry if I was a little bit hesitant at the start of my talk. It's actually really hard to give a talk from home, but I was really happy to be invited and share some of these results with you. What I've shown today shows work from my lab over a number of years. Particularly the screen was done by Hector, Irene and Florian. The histamine mutant was characterized by Florian almost solely with help from the rest of the lab. I've shown really beautiful work from Matt Parker and Matt Winter, and you'll be hearing from Matt Parker later on today. And then other members of my lab, Palumi, Kenwin, and Elisa also helped out. And I'm grateful to have funding from the BBSRC, who funds Palumi, and the NC3Rs funds our Animal Welfare Project. And I'll be happy to take any questions that you have. Thank you very much.